for 2025. Paul, thank you very much.
So let me start. I have this kind of overwrought title, right? Developing therapeutics that use the immune system to eradicate cancer, and we will talk about that a little bit. Here are my disclosures. You know, the title I really like is that it's all biology, right? So, you know, what I'll tell you about today is a biology story that started here. Actually started when I was about 7 and started birding, and became really a habitual naturalist, and I still am today. So Bill's lab and the then Department of Zoology were a really good fit for me because I was just that kind of kid, right? Importantly for me, from a scientific thinking perspective, Bill had a really clear view of some questions in research that he wanted to address. And he had the technologies in the lab to do it, and he had the scientific rigor to direct people like me, who were newbies, in this exercise in hypothesis testing. Equally importantly, he had really robust collaboration network of equal-minded PIs, right? And most importantly, he had grant funding. And so I showed up, I worked in the lab for a while, and I was immediately out in the field in Mexico. Bill was a active mentor, and I can't stress how important this is. Understanding that, you know, a lot of times you don't know what you're doing and you need to be shown and taught how to do. It's a really, really important skill. So happily, you know, and as we've just heard a little bit, right, this exercise in traversing Mexico and collecting Peromyscus and samples derived from Peromyscus. Here are just some of the, you know, this landscape is spectacular, you know, and you're in the mountainous regions. And the hypothesis was really simple. This kind of geography ought to isolate populations that could become distinct species, right? That's the driving hypothesis. And, you know, it not only kicked off my career, but it, as I read last week, it kicked off a gigantic literature spawned by Bill and others on really understanding this entire genus, right? So it's a lot of work and a lot of evolutionary insights. And it's where I started. So we were working on Peromyscus boylei, it's a cute little brush mouse. The other part of, you know, what we did is it's not just biology, but it's also technology, right? So this was in the 80s, you know, and we were using what were then considered modern techniques, right, to uncover species relationships molecularly. Importantly, and this turned out to be really important for me, we were quick to publish our findings, right? So that when it was time for me to leave and go down to Boston and try to break into the larger, you know, sort of biomedical, corporate biotechnology field, I had a story to tell people. So I didn't just show up with a list of techniques that I knew how to do. I had tested hypotheses using that technology, derived answers, and put together a narrative that I could share with people. And that was a really great skill that I learned really early from Bill. And I kept that skill with me throughout my career. So careers are funny things, right? They can go really well. They can go straight up. Sometimes they can go straight down. Mine went all over the place. So I moved to Boston. I joined the Howard Hughes Medical Institute, which is a very heavily funded, posh institute. It was at MGH. And I was hired to do molecular biology, which I knew a little tiny bit about. And I worked on NGF, which I knew nothing about, right? So it tells you a little bit about the importance of the technology, right? Once you have these techniques in your hand, you can apply them to different questions. At that lab, I wasn't there very long, but at that lab, I learned a lot of molecular cloning techniques. I learned what a restriction endonuclease was, right? Things like that. And we did a lot of gene expression analysis. So it was a different sort of field of molecular biology than what I had been studying. We did pretty well. We wrote up some papers, but the Hughes pulled our funding about a year later. I ended up in line at the Mass Department of Unemployment, right? So, you know, none of this stuff is guaranteed to have a great outcome. So then I jumped the river. Mass General was on the Boston side, and Kendall Square, which is sort of the birthplace of biotech, is on the Cambridge side of the Charles River. So I crossed the river And I got a job, believe it or not, as a molecular biologist, of which I still knew really relatively little. I had 1 year at the Hughes doing molecular biology techniques, but I didn't consider myself a molecular biologist. But I was hired at Repligen, which at that time was a growing company. And I got really lucky because I was handed by Gary Gray, another really important mentor in my career, to run two collaborations, one with a lab at Dana-Farber that was then studying the new field of T-cell co-stimulation, and another one with a group at the Naval Medical Research Institute in Bethesda that was expanding T-cells to treat HIV patients. The goal was patients are T-cell deficient, can we expand the patient's T-cells and give them back? It was a great idea, it didn't work because this was before we knew CD4 was a co-receptor for HIV. So we were just giving the patients a huge virus depot to expand in. So it didn't work. Nonetheless, right, we pushed forward our programs, got to the point where we're doing bona fide drug development. And so this was my first exposure to what GMP meant, how to file an IND, all those kinds of things that become important later when you're developing drugs. And biotech funding back then, much like it has done today, collapsed, right? And so the company just imploded. Most of the employees were let go, and I ended up back on the unemployment line. But I wasn't there for very long, and I'll show you why. And importantly, this type of work, right, working on T-cell, B-cell interactions, working on the immune system became the foundation for everything I did subsequent to that. So even though this didn't end well, it was a really important, stopping point. And I was there for about 5 years, so it's not like it happened fast. So then, after I left Repligen, I was hired at Biogen, which at that time was a struggling biotech. And I was hired into a lab, and they hired me based on the collaborations I'd done at Repligen. They hired me as an immunologist. I had studied no immunology ever, right? And I was flabbergasted. And Paula Hockman, again, these are, really excellent people in my development. Paula said to me, well, no one understands this stuff, so don't worry about it. I'm like, that's what the molecular biology guy said, as general, right? So, I think everybody back then was just learning. And like now, the techniques change so quickly that you almost can't be behind because everybody's always behind as new technology comes out, right? What it does say is you've got to be nimble, and you've got to be willing to change, your cap, right? So now as an immunologist, and I stayed an immunologist for about 15 years. I stayed too long. So for those of you who go into industry, you definitely don't want to stay at one place for a long time because your career will grow as you move between companies and gather different experiences. So I didn't know that at the time, and then it was like sort of too late. But I got to work on a bunch of drugs, none of which I invented. So these are all immune system drugs that are designed to treat autoimmune disease and chronic inflammation. And I worked on a whole bunch of things in that area. But the biology that I discovered and spent most of my time on was lymph node development, Peyer's patch development, organization of immune organs. And this organization, you can see it over here. There's like a B cell, there's a B cell zones, and there's T cell zones. This is a mouse lymph node, inguinal lymph node stained for a B cell marker and a T cell marker. This organization is critically important for how the immune system actually functions. So these different cell types, dendritic cells, B cells, macrophages, T cells, come together in a highly regulated manner to induce an immune response. And that's something I spent a lot of time studying. So at that point, Biogen was changing trajectory and becoming a neuro company based on the MS franchise, right? So Biogen thought, well, wouldn't it be great if we can expand our MS work into neural pathologies more broadly? So they wanted to go into things like stroke and Alzheimer's and Parkinson's. And I just couldn't, I just didn't really feel like I could pretend to be a neurologist, right? Like it wasn't a good fit anymore. And also 2 of my projects during that transition, 2 of my projects were cut. So one of them was an immuno-oncology program. This was on a putative immune checkpoint molecule called TIM1. Putative, I say putative because it turned out that it was not an immune checkpoint molecule. It's actually a receptor for phosphatidylserine. So it's really just a scavenger type receptor. But we had run this project, we'd gotten some interesting data, we'd written up some patents, and a venture capital firm called MPM Capital in Boston wanted the project because they were putting together what was likely the very first private immuno-oncology company. So they went back to my friends at the Farber and licensed out a PD-1 and a PD-L1. They got a TIM-1 from me. They got a TIM-3 from a guy at the Brigham and so on. And they licensed in all this IP to put a company together that would work on immuno-oncology. I joined as a consultant. So I quit Biogen and went to MPM to help them put this company together. That company was bought very quickly by Novartis. So I just want to make the point that if you're going to do this kind of work, try to get equity. Because Novartis paid a mint for that company. I didn't get a penny out of that, even though I'd helped put the company together. So that was a stepstone into working in the immuno-oncology space, right? So I'd been in immunology, and it wasn't a great stretch to say, well, this guy knows some immunology, so let's put him to work on other targets in immuno-oncology. And I put a consulting firm together to work on that for a number of years. And the consulting firm was designed really so that I would keep going out, meeting different people, networking, and eventually, I used it to found 3 other companies, all of which failed because I couldn't raise funding, right? So I kept trying, and eventually the CAR T-cell therapy field exploded. And this is a subset of immuno-oncology where instead of messing with the immune suppressor molecules on the T cell, you're actually taking the T cell out of the patient and transducing it and putting it back in. And I'll show you what that looks like in a second. But it is IO in a sort of fundamental definition of IO. It includes T cells. And one of the things that was happening is we were seeing data where these CAR T cell therapies were doing really well in certain diseases. We're also seeing that the cost of these therapies was really outrageously high. So it was worth it because for a subset of patients, they were effectively being cured, right, of advanced blood cancers. So we took a look at this space and thought, well, we've been building biologics for a long time. CAR T cells are really complex. Can we make biologics that will help CAR T cells do their job better without having to make a whole new CAR, right? So that was the premise for Aleta. And when we raised seed funding, my co-founder, Roy Lobb, and I raised seed funding and started the company, opened the lab in early 2016. So just, I won't go through this in any detail, just for people who don't know about CARs. This is a therapeutic modality where you remove the T cells from the patient. So the patient is apheresed, the T cells are isolated from the peripheral blood mononuclear cell population, and then they're transduced with a gene that puts this thing on the side, this construct on the side. It puts that on the cell surface. So what that is an antibody domain to a tumor target antigen linked to co-stimulatory domains that activate T cells. So when that T cell sees that antigen, that T cell becomes cytotoxic and kills whatever is expressing the antigen. That's how cell therapy works. So you have to take the T cells out, transduce them with a lentivirus, expand them, put them back in. Takes about 3 or 4 weeks, costs about $500,000 per patient. So therapies like this, in order to have bona fide healthcare value, have got to work really well, right? Now, happily, the only place you can get a CAR T cell therapy in the state of Vermont is right here. So if you want to learn more about CARs, there is a group right here at the medical center working on some of these CAR T cells. They're broadly approved in 2 therapeutic areas. I'm just showing you one, which are the B cell malignancies, right? So acute lymphocytic leukemia, ALL, and the large B cell lymphomas, right? So these are dreadful diseases when you fail standard of care. Many patients can be cured with standard of care, but for patients who are not cured, their outcomes are very dim. The other set of CARs that's directed to a different antigen, not CD19, are for multiple myeloma. So those are probably the two indications which the medical center here is treating with CAR T-cell therapy. So, what are we trying to do to improve CAR T-cells? And I want to show you some data using published clinical data on axi-cell, which is this guy, Yescarta. It's called axi-cell for short. Using axi-cell data as an example. So this is like both the good news and the bad news. So this is early data. So these are third line plus advanced lymphoma patients. Third line plus means they failed at least 3 prior lines of therapy and they've relapsed. And the outcomes for those patients are really grim. They're mostly, their overall survival, median overall survival is probably less than 2 years. Small subset of them do really well on allo transplant, 5 to 10%. So these same patients now are in a cohort that are given axi-cell, the CAR T-cell therapy, directed to CD19, which is expressed on the surface of the lymphoma cell. And here we are 5 years later, and 40% of these patients are still alive. If this was standard of care, the number of patients still alive would be near 0, right? So this is a transformative advance in the treatment of this blood cancer for about 40% of the patients who get it, right? Leaving another 60% of the patients not getting that benefit. Now, this progression-free survival data, I like to use it because it shows over here, it shows this really steep curve after therapy. And what that's illustrating is that there's about 10% of patients who don't respond at all. So they fall off the cliff. There's about 80% of patients who initially respond, and of those, 2/3 of them then relapse, and they relapse really fast. So you can see that this progression-free survival curve starts to flatten out at like 4 months. So whatever is going to happen has happened, and it happens fast. So we took a look at this data at ASH, the hematology meeting in late 2016, and we came back and we thought, well, we're creating this platform of biologics. Can we use this platform to address some of the causes that underlie the poor responses? In other words, can we help the 60%, right? Because the 40% are doing really well. So we took a look at what's driving these poor responses and these relapses. And really 2 big things jumped out. One is that CD19 antigen density in lymphoma is really variable, and you can see that over on the left-hand side here, right? So look at diffuse large B-cell lymphoma. It's a log 2, but you've got patients who are way down here, and you've got patients that are way up there. And it turns out that the patients that are low for CD19 expression, before they receive the therapy, their clones escape, right? And you can see that if you look at progression-free survival over time. If your lymphoma is CD19 high expressor, you do about 20% better in terms of the duration of progression-free survival than if your lymphoma is dim. And that doesn't mean every cell, right? That's an MFI. It's like a mean fluorescence intensity on the lymphoma population. You just have to have a subset that's dim because they'll escape, right? Because the antigen density is too low to trigger the CAR. So that's resistance because you have a pre-existing cell pool that won't be targeted by the therapy. That's resistance. Relapse is driven by active loss of CD19 post-therapy, right? So your CD19 is present. So here's a patient visualized on the left with PET CT, and they have a big lymphoma mass in the leg. It's CD19 positive by immunohistochemistry. So that's what you're attacking with the CAR T cell. It's going to bind to all that CD19 and kill all those lymphoma cells. 1 month later, the patient is in a partial response. So you can still see there are some lesions that are still PET positive. So this patient still has lymphoma. At 3 months, now they're back in progressive disease. Their lymphoma is growing avidly. You have large masses and many small masses, and that large mass, when it's biopsied, is now CD19 negative. So the lymphoma, this is cancer resistance 101, right? If you target something on a cancer cell, that population, which is gigantic, highly proliferative, and mutagenic, finds a way to escape expressing the target for the therapy. And I won't go through, this is just the types of mutations that have been seen, shown in the middle. The number that's important is on the right. So in a couple of cohorts, one at Stanford, one the French consortium, looked at their CAR relapses after an initial response and quantified them with respect to CD19 downregulation and identified that up to 60% of their relapses were due to loss of CD19. So you have these two things going on. One, you may have a clone that's dim to begin with. And then secondly, you might be uniformly CD19 positive. And you have these relapse mechanisms, both of which contribute to that 60% who don't derive benefit, right? So you need a lot of CD19 expression, and you need that expression to be persistent so that the CAR continues to work. So T cells, when they get activated, activated T cells, if they're not continually activated, they self-destruct, right? Because an activated T cell is a dangerous cell. That's how we get autoimmunity, for example, or chronic inflammation. So they're designed to shut themselves off if they're not continually getting stimulated. So that the antigen's got to be present, and it's got to be persistently present. Okay, so what did we want to do, right? This is a sort of long intro, but what did we want to do, right? We asked a simple question: can we improve the antigen density, and can we stop antigen loss? And the clinical goal is pretty obvious. We wanted to move this curve up, right? So more patients are receiving durable remissions. And we had a third goal, which made us a little bit unpopular with the venture capital community, which was to make it cheap and easy to deliver, because CARs are expensive and hard to make and hard to deliver. And we didn't want to put another $500,000 solution on a $500,000 problem. Now you've got $1 million therapeutic. That might be fine in rare diseases, right? Pediatric rare disease, things like that, where you're one and done. And something like this, there are way too many patients to make that kind of math affordable. So we invented a small biologic. Our platform was called CAR-T engagers. We were also doing T-cell engagers. CAR-T engagers are pretty simple molecules. They have the CD19 extracellular domain. We had to mutagenize it to make it expressible, protease resistant, and thermostable, but we did that. Now it's just a protein that you can express, right? And we linked it to an anti-CD20 domain from an immunized llama. So we like llamas because their antibodies are single domain heavy chain antibodies. They're really small and they're really stable, whereas mouse antibodies, SCFEs, things like that, are neither small nor stable. So we like the llama. And this small protein just does two things. It binds to the anti-CD19 CAR domain. So remember, what's put on the cell surface of the CAR is an antibody domain that binds to CD19, which is that protein that we put in this fusion protein. And then the anti-CD20 binds to a second antigen on the lymphoma cell, right? So lymphoma cells are 19 positive, 20 positive, and a bunch of other things. 20 is really stable. It's really bright. All we've done here is paint CD19 onto the lymphoma cell by binding it to CD20. And this is a really high affinity binding event, down around about sub-nanomolar, about 0.7 nanomolar. So once it binds, it stays. And then the CAR T cell can't tell the difference between natively expressed CD19 and CD19 that we've painted onto the cell. The CAR just sees CD19 and it's triggered and goes off and it kills the lymphoma cell that it sees. So just to lean into this protein just a little bit more, we did add an anti-albumin domain. Albumin is recycled through FcRn just like Ig is. So when you add an anti-albumin binder, it binds to albumin and it gets recycled. So that gives you half-life extension. That's just a little trick that we played. We like this drug a lot. It's small, it's easily expressed, it's really stable, and the cost of goods was really low. So we have a cheap drug that we can vial and we can ship anywhere at -20. So then we needed to raise the money to make the drug at scale, so at GMP. So we did a Series B. Our Series B was successful because we had won a grant in the UK from Cancer Research UK to run a trial. in diffuse large B-cell lymphoma patients who had already received a CAR. So when you have that kind of a pitch, right, to venture, you say, well, I'm bringing in about 40 million in non-dilutive funding for the clinical trial, and I'm only asking you for 15 million to make the drug. That's the kind of pitch that they like. So we were able to raise the B and then go make the protein GMP. We ran it at a company, at a contract development and manufacturing company called Wuxi Biotech in China. We made about 15,000 doses of the drug in 500 liters. So if you're a CMC guy, like those numbers are really good. You know, you're not doing 20,000 liter bioreactors. You're doing 500 liters, and you're getting enough drug to fund multiple clinical trials, and it's really stable, so you don't have to make it again. So all that was really promising. Just some POC data, just to show you the mechanism of action. So this is a lymphoma cell shown in the FACS plots here, where we CRISPRed out CD19. So it's CD19 negative lymphoma, still CD20 positive. In the middle is a bead count flow cytometry. So we're actually counting the number of receptors on the surface of that cell. And what you can see is that there's 250 plus thousand copies of CD20. And when we add Aleta 001 to that CD19 knockout, we get the same number of copies of CD19 apparently expressed on that lymphoma cell. That just means they're bound to CD20. So the flow cytometer sees, you know, the binding event, sees that there's CD19 bound to that cell. So it follows that if you try to kill that CD19 knockout lymphoma cell with a CD19 CAR T cell, it won't work. And so the 48-hour cytotoxicity is 0% because there's no CD19. But then if you add Aleta 001, which is the name of the drug, that binds to CD20 and expresses CD19, puts it on that cell, you kill all of the lymphoma cells in 48 hours, right? Now that's the proof of concept experiment, because you're going from a negative, like there's no CD19, and now we're putting CD19 on it, and now we're killing it. But the other part of the argument is that we want to make antigen density brighter, right? So this is the wild type. This is a wild type lymphoma cell. That's CD19 and that's CD20. People forget, right, that flow, you know, is a log scale, right? That MFI is a log scale. So when you do the math, it turns out that there's about 25,000 copies of CD19 on that lymphoma cell. It's shown in orange. And then again, you know, there's 200,000 or 175,000 copies of CD20. So if you add them together, guess what happens, right? Now the apparent density of CD19 is the addition of the natively expressed CD19 and the CD19 that you've painted onto CD20. That's why the blue line at the top is bigger than the red line, which is the CD20 native expression. We're adding more antigen onto that lymphoma cell. We're making it super bright. So does that have an impact on killing? In order to see that, you have to actually tinker with the killing cytotoxicity assay parameters. So we dropped the effector target ratio, we added less CARs, and we ran it for a shorter period of time. And you can see that if you add the CAR now to this cell, right, you get 25% cytotoxicity at 18 hours. But if you add Aleta 001 on top of the CAR, now all the cells are killed. So you boosted the ability of this CAR to kill, and to kill quickly. And in lymphoma, that's really important, because lymphoma is a rapidly proliferative malignancy. And patients who've relapsed from standard of care, the clinicians tell us that for these patients, every week counts, right? So you can lose a patient in the course of a number of weeks because this is so aggressive. So when you put the CARs in, right, you're asking them to race against the lymphoma and win. They have to proliferate while the lymphoma is proliferating, and it helps that, as I mentioned, T cells that get stimulated want to keep seeing antigen. So these guys are serial killers. Each CAR T cell will kill dozens, if not hundreds, of lymphoma cells. That's the only way the CAR wins the race. So to win the race for more patients, you want to kill more quickly at lower effective target ratios. So you don't need so many T cells. So this was gratifying data. We took this whole, I'm not going to show it for time considerations, but we took this whole knockout system into a mouse and showed we can kill in vivo. And we did all the IND enabling studies, and that led us back to the clinical hypothesis, which I mentioned earlier. We can increase the durable responses and cures for these patients by preventing the poor responses due to CD19 dim lymphoma cells and the relapses due to active downregulation, the two causes of treatment failure. There are others. These are the two major causes of treatment failure that are tied to CD19 expression. Okay, so then we went to the clinic and we've started a phase 1 and we're about 12 patients in. And what happened, with a phase 1, you have to start with a low dose, right? So we started at 0.4 mg per kg, we went to 2 mg per kg, and now we're at 6 mg per kg. In the 0.4 mg per kg dose cohort, which is the lowest dose, we saw pharmacodynamic proof of response. So the CAR vector copy numbers increased over a couple of cycles. That means the CARs are expanding after you've given the protein. The CARs are now seeing antigen again, right? And the CAR T cells in the periphery jumped to up to 40% of the cells circulating in the blood. So the majority antigen being seen by the T cell population is now the lymphoma antigen CD19. In the higher dose cohorts, 2 and 6 mg per kg, we've also seen clinical responses in patients who were in the midst of relapsing with progressive disease. So we've had patients who are in a progressive disease and now they're in either long-term partial responses or they're in complete responses, which means we've turned their fate around because they were gone, right? And now they're not. So the POC is here and we feel pretty good about the early clinical data. We need more clinical data, obviously, in order to prove the point, but the early data are really promising. Okay, so that just says to me that the technology works, right? We've taken the risk out of the technology platform because in our lead program, we're seeing proof of mechanism. So that's great. Now we can ask the question, what else can we do with this? And we stuck with CD19 targeting CARs, and we've decided to redirect them to other indications. And the reason is that CD19 CARs work really, really well compared to other CARs, right? Even the BCMA CARs are not as effective as CD19 CARs. Importantly, the reason for that is that they also see and deplete the normal B cell pool, and that is a persistent, self-renewing source of antigen that's independent of the lymphoma, right? The bone marrow is pumping out pre-pro B cells every minute of every day. This CAR never runs out of antigen because it's got a normal B cell pool. And not only is it self-renewing, but it's immunologically relevant because they're B cells, and those are cognate partners for T cells. So even though the interaction is fatal for the B cell, it's productive for the T cell, chemokines, cytokines, adhesion molecules, and so on. None of this happens when CARs are directed to solid tumor cells, and we've had, a decade of failure in making CARs to solid tumors across indications. You get occasional responses. They're not very durable, and the reason turns out to be that solid tumor cells, unlike B cells, they provide there's no active embrace with the T cell. In fact, they're deliberately immunosuppressive, and they have none of the immune system tools that a T cell wants to engage in order to be a happy, healthy T cell. So we thought, all right, well, we've got these CARs and we have this CAR T engager technology. Can we use the CARs and just give them another antigen to see? Let them still see B cells so that they're proliferative and they're fit, but then redirect them to other tumor antigens in solid tumors. So this is a program where we still have the CD19 protein, same thing, right? Binds to CD19 CAR T cells. So that interaction hasn't changed at all. It's just CD19 and the CAR. But on the other end, we've got a binder to HER2. So HER2 is a solid tumor antigen notably expressed in sarcoma, breast cancer, gastric cancer, bladder cancer, non-small cell lung cancer, and a host of others, right? So really important, you know, antigen in solid tumors. If any of you have heard of HER2 or trastuzumab, these are antibody therapies directed to HER2. So we thought, well, we can let this CAR T cell population, you know, be energized off the normal B cell pool and also let it go see HER2 and eradicate HER2 positive tumors. So I'm just jumping straight to the mouse model this time because I showed you guys, you know, in vitro data before. On this program, we took an ovarian carcinoma that's HER2 bright, and we put it in an immunodeficient mouse. These are NSG mice that are really immunodeficient. And after about a month and a half, you know, all the control cohorts all die, right? There's a lethal carcinoma in this mouse model. So all of these at the top, they're all gone. The mice that got anti-CD19 CAR T cells and this HER2-directed CAR T engager cleared the carcinomas and they all survived. So now we know, right? Translationally, mouse models are not, you know, everything works in a mouse, right? So I don't want to oversell the data, right? But it at least says that conceptually, we can take an anti-CD19 CAR T cell and send it after anything we want. So we thought, all right, well, that's cool. This program is going forward. We're attempting to drive this program forward as a collaboration with one of the major hospital groups in the UK called Guy's and King's in London. So we thought, all right, well, that's good, but, you know, can we make the CARs better? You know, just letting them see B cells may not be enough. Can we make them better when they encounter the solid tumor cells? So we went back and we decided to add co-stimulation ligands to the CAR T engagers. We work on two. One is CD58 that activates CD2. I'll show you a little bit about that. We also worked on B72 that activates CD28, and that was a construct I first made back at Repligen in 1997. It's one of the papers I showed earlier. We're really informed by recent data in the field using molecular imaging studies to look at how the T cell surface membrane changes once that T cell encounters antigen, right? So we always had this concept that there was this T cell receptor zone and everything happened underneath it. You know, all the signaling went downstream. That's not true. That's how the TCR works. But the co-stimulatory ligands actually have to move. And they move into a corolla. It's shown in pink in the Michael Dustin paper. They move out from under the TCR. That allows the T cell receptor to relax and allows the co-stimulation to continue. And engagement of the T cell receptor has to be punctate, otherwise you overstimulate it. So this is a really clever evolutionary trick that, you know, the cell membrane has evolved to move things to where they want to be. And I think it's one of the reasons that antibodies that are used to trigger CD28 don't work very well, because I don't think they let the proteins move where they need to go. Whereas we use G4S linkers that are really flexible, so the proteins can go wherever they want. All right, so that was our hypothesis. We're going to use the B cell basis for the CAR. We're going to add co-stimulation to the CAR T engager. And we put this molecule together. So this molecule is, this is a program I really want to work, right? So these two antigens are expressed in a number of indications, but prominently in the pediatric CNS cancers. These are typically stem cell cancers that arise from different geographies within the CNS. They're deadly in children. They're deadly in adults too, but they're really nigh impossible to control in the pediatric setting. So there's a tremendous unmet need. And we thought, well, let's just put this molecule together and let's give it our best shot. So we have two different VHs, one that binds B7H3, one that binds IL-13 receptor alpha 2. And we have the CD19 engager for the CAR still in the middle. So this is still a CD19 CAR program. We're not making 5 different CARs. We're just making 5 different proteins. It's way easier. And we've added these 2 little CD58 co-stimulatory domains that bind to CD2. You have to add 2. I don't know why, but you know, CD2, CD58 binding is low affinity, high avidity. So it sort of makes sense that you would need a couple of copies of it in order to get active binding. What happens is really cool. So this molecule now, everything I've been showing you has been about binding to the tumor cell, right? Can the CAR T engager bind to the lymphoma cell? Can bind to the HER2 positive carcinoma cell? Here, we're also asking, can the CAR T engager bind to a T cell, right? So these CD2 domains will let the CAR T engager engage the T cell. Now you're engaging both cell types on either side of that interface and making a really productive kill zone for that CAR T cell. And what happens is the killing jumps quite dramatically. We're now down to the nigh 1 picomolar range, right? This is a scant amount of protein. The reason it happens is that engaging CD2 drives gamma interferon secretion from the T cell, and gamma interferon is a key killer cytokine for cytotoxicity. So it's what's driving killing of the tumor cell. So we're weaving, the control here, I'm sorry about that, the cartoon, the control here, CTE006 doesn't have the co-stim domains. Co-stim CTE006 has the co-stim domains. So you get T cell binding and you get much higher gamma interferon secretion at much lower concentrations. The concentration of protein here needed to induce gamma interferon secretion is very, very low indeed, and that helps kill. This program, I think this program is best suited to foundational support. So we're trying to pursue the disease foundations that specialize in pediatric cancers to provide support for this program. What it requires and what's happening in the field, because CARs are so expensive, individual hospitals and institutions are making their own CAR-19s. The patent landscape there is open, right? So you can go back to, the original CAR patents and they're expired. You can go make that CAR-19 yourself tomorrow, and you have complete FTO because those patents are expired and those designs are now in the public domain. So hospitals, and Barcelona is notable for this, UCL, the big centers in Brazil, they're all making their own CARs because they can make them much less expensively than the $500,000 that J&J or Bristol Myers or Kite Gilead has to charge for their CARs, right? So that's the kind of setup you need. You go to an institution that has their own CAR, you get the disease foundation to help make the protein, which isn't that expensive, right? 10 million maybe. And then that's your study design. And I think that's the path forward for a program like this that's not going to get a lot of institutional investor support. Okay. We have a number of these programs. I talked about 002, which is the HER2 program, and 006, which is at the bottom. There's a couple others I won't go through. Happily, because these are proteins, we can be in the clinic with these proteins. We can get them GMP manufactured and IND enabled in around 15 months. So we could be in the clinic in a year and a half with an institution that has an anti-CD19 CAR. So this is where Guy's and King's in the UK comes into play because they're developing their own CAR. And these are the kinds of things they want to do. All right, so I started off by saying, this is all biology. And I think, this story is about following the biology to whatever, either discipline or disease indication or immune function that it leads you. It led us to develop a new class of biologics called CAR T engagers. And we're now driving, the first clinical program in lymphoma patients, and we're trying to broaden the technology by adding some of the tools, that we stumbled across decades ago on how to stimulate T cells productively and in a way that's organic, right? With using the native ligands to tickle the T cells and provide them with signals that they need for robust activity. Just a couple of shout outs to the group at Biogen. Emma Anderton runs the program within Cancer Research UK. That's the Aleta 01 program. Sridhar Shiganti is the clinical lead and is at the University of Birmingham in the UK. And at the bottom are some of our support from our venture partners listed at the bottom. Okay, that's it for me.